welcome to yet another revision stream. We're on a roll with these now. We have revised so far carbohydrates, proteins, lipids. The other day we did ATP, water, and inorganic ions, which only leaves two things left, DNA and RNA, which we are doing today. So I would argue DNA and RNA are the hardest topics. DNA is quite easy, but RNA comes with the lovely inclusion of transcription and translation, which I would argue they're understandable, but you have to wrap your head around a few little things that I might have forgotten. So what we'll start off by looking at the structure of DNA, all of that, and then we will move on to RNA. We also need to talk about DNA replication too, because I always forget that's a thing, but it is a thing that happens. Next week, just as a little warning, I guess, we'll be looking at things like mitosis, meiosis, structure of cell membranes. The week after, we'll be going on to mass transport, which are things like digestion, absorption, gas exchange, the circulatory system. And then that'll basically leave us with, I think, 2 more weeks where we'll be able to revise anything we want. So any topics that we're struggling with, any practicals, that we have 2 weeks before the exam where we can just cram in as much revision as possible. So let's get on with this, shall we? So the structure of DNA. I'm going to write the word DNA because in an exam, a lot of people seem to get very caught out here just by how simple DNA is. Deoxyribose nucleic acid, okay? Deoxyribose makes DNA slightly different to ATP. There's been many exam questions where they'll talk to you about, compare the structure of ATP and DNA. People waffle about all of this stuff, about covalent bonds, whatever. One of the easiest marks to get is the fact that in ATP it has ribose sugar, in DNA it has deoxyribose. So let's draw this structure, shall we? So firstly we're going to start off with the pentose sugar here, which is obviously going to be deoxyribose. Then we're going to draw our inorganic phosphates, and then next to that we are going to have our nitrogenous base. Let's look at the base quickly because it can either be ATCG, we got adenine, thymine, cytosine, or guanine. So deoxyribose, inorganic phosphate. Couple more things to note are the bonds. There are two main. I keep forgetting to pop chat on. It's fine, I'm sure. Where is it, pen here? There are two. Well, I don't seem to recall whether this is an ester bond or not, so I'll look into that in a sec. But this is a phosphodiester bond. This is a hydrogen bond. Because that connects the two complementary bases together. So that is the structure of DNA right there. There are 3 properties of DNA that I misread these a lot. I will try and remember them, but I might need to check online. One of them is the fact that DNA is degenerate. Also, the idea of DNA is that it's a very stable molecule. But one of them's degenerate, and I don't recall the other 2, which is annoying because there was a exam question worth like 6 marks where you would have got it for knowing the definitions of the properties. Degenerate, by the way, all that word means, I'm 99% sure, is that the same, I'm pretty sure it's where more than one base sequence can code for the same amino acid. So if we had like a combination of like AGT and there was a mutation which led to that, both of those might code for the exact same protein nonetheless. Because it's degenerate. I'm 90% sure that's what it means. But I'll have to check. I love all the buns. Okay. I get that, buddy. I get that. So that is something I'm gonna pull out my laptop for, because I gotta make sure that I know these definitions. What we'll do though, I'll go on the AQA website. We'll look at the question first and not the mark scheme, just in case. A-level biology past papers. Question paper for A-level. I think this is it? Okay. Yep, I'm 90% sure this is the paper. Oh, I can't find it. Shit. I don't recall what paper it was on. Was it on AS biology? If it was on AS biology, that is evil. Maybe it was. It was! Define universal, non-overlapping, and degenerate. Okay. Universal and non-overlapping. Those are the three properties of, I guess, more genetic code, but we can talk about it in relation to DNA. I guess the terms are quite interchangeable. Okay, so degenerate we know is where multiple bases can code for the same amino acid. Or I guess it would be multiple triplets, because obviously DNA works in triplets. So I'm 90% sure I have this one right. Universal. Does that just mean that every organism has the same 4 bases? And what, non-overlapping, does that mean that, non-overlapping in relation to DNA, genetic code, does that mean that only 3 bases are read at once? Like, only one codon is read at once, maybe? Let's have a check, shall we? Universal is where the same codon/triplet always codes for the same amino acid. Okay, interesting. So let's actually write down these definitions so we know what they're on about. So universal is where the same triplet always codes for the same amino acid. So I guess I was kind of along the right lines when I said every organism has these. I should have just looked into it deeper. Every organism, if this was A, T, and G, in every organism that would code for the exact same amino acid. Universal, we've done. Non-overlapping. Each base is only part of one codon. Okay, so I was, again, kind of on a similar line there. I said that each triplet is only read once. So what it means there is each base is only part of one triplet. So if we say A, T, A, C, G, A, it means that that A is not going to be part of that triplet as well. It's separate. So that's a nice easy one to remember, I think. Just each triplet is only part of one triplet. Each base is only part of one triplet. I think that's an easy enough definition to remember right there. So sorry, that's universal. That's non-overlapping. Degenerate. More than one codon codes for each amino acid. Okay, that is what I was trying to say, but I couldn't word it right. So yeah, more than one codon. More than one codon codes for the same amino acid. So, those are some very interesting definitions that, honestly, we have not been taught. We have not been taught those in our classes. The only reason I know them is because I've done the mark schemes. I've looked at the mark schemes before and the exam questions. So that right there, how many marks was that? 3 marks. That right there would have probably made everyone in my class drop 2 marks. Maybe even 3 if they couldn't describe degenerate properly. That is mental, because that is one of the first questions in the paper. And if you know for a fact that you don't know the answer, that's going to mess up your kind of flow, and you're probably going to get a lot more wrong as a result. So, non-overlapping is where only each base is only part of one triplet, not multiple. That's easy enough to understand. Universal is where the same triplet always codes for the same amino acid. And to add on to that, degenerate is where there are multiple triplets that code for the same amino acid. So they go hand in hand. It's easy to understand. Oh, I would have gotten non-overlapping right, because it says non-overlapping, bases only read once, or triplets only read once. So I would have gotten this. I would have gotten that right. So I would have gotten that mark. I might have gotten that mark. I would not have got universal. So that right there, I might have gotten 2 marks max. That is probably still 1 more mark than what anyone else could have got in our class. And that is why we need to revise this, because if the teachers aren't helping us by telling us everything, how are we going to get all the marks? So that is DNA. Let's move on to DNA replication, which I don't recall fully. I do not recall this one. So let me try and do it off the top of my head, and we'll see where we go. Okay. I'm just going to draw it like this, because I am too lazy to draw it properly. We're going to assume that all of these are bonded, like it's combined to begin with. So I think the first step is we have the action of DNA helicase causes the DNA molecule to become unzipped. Now, I don't know if you're allowed to mention hydrolysis in an exam, but it should be a hydrolytic enzyme from what I remember. But I think you have to say that it breaks the bonds. It breaks the hydrogen bonds. Hydrogen bonds are broken, causing the molecule to unwind, or unzip, I guess. Should we just say unzip/unwind as H bonds break? So that's the first thing right there. The DNA helicase causes the molecule to unzip because the hydrogen bonds break. Next, I am 90% sure that we have our free-floating bases, which I'm just going to draw with like little things like that. Free-floating bases will bind to their complementary base. So A, T, C, G, apples, that would be T, thymine, adenine. You get the idea, okay? That's where they would bind. They would bind to their complementary base pairs, and that's the second idea. So these free floating bases bind to complementary base pairs. Or I think bind could be a little deceptive because they're not bonded. So should I say line up to or go to? free floating bases go to complementary base pairs. Then, once we have all of these together, like this, we then have the action of DNA polymerase. And what this does is it creates the phosphodiester bonds between the adjacent bases. Okay, hold on. Free floating bases bind to complementary base pairs could be right. I'll leave it in for now. DNA polymerase catalyzes the phosphodiester bonds forming between adjacent bases. And from that, we have our 2 new strands of DNA. It's called semi-conservative. Because I think the idea is that each one is half the original. Okay, look, we've split. Right, I'll demonstrate it with my pens because I have a lot of pens. I just ignore the colours. Imagine you're colourblind, right? Imagine this is a single DNA strand. We are literally pulling them apart like this, and then doing that. So now each DNA strand is half the original, okay? Half the original. And we've replicated the DNA there. DNA replication, I don't exactly recall the reason for this, but I know it happens during mitosis, during interphase, I believe, during growth cycle 1, if I'm mistaken, maybe. So there you go. I think that's everything for DNA. I recall there was a practical, or like an experiment that took place related to DNA replication. Don't know who by, but I'm pretty sure they got light and heavy nitrogen. And because of that, they were able to figure out that as time went on, the molecules would get lighter and lighter until it reached like a balance, because they would become half as light. From what I remember. So anyways, should we go do a quiz on this to make sure I haven't forgotten anything? I think we should. Okay. Yeah, it's the Mielson and Stahl experiment. Okay, cool. Right, 20 questions. Let's go through these. During semi-conservative DNA replication, one separated each strand of a DNA molecule acts as a template to which complementary free-floating nucleotides. Shit. Okay, free-floating nucleotides bind to their complementary base pairs. That makes a lot more sense. Cool. DNA polymerase forms what kind of bond between adjacent nucleotides? That's going to be phosphodiester. During DNA replication, DNA polymerase forms bonds between adjacent nucleotides in the new strand being synthesized. Which of the following is not a nucleotide? Whatever that one is. Which of the following is not a base found in DNA? Guanosine. What the hell does that even mean? Which of the following is complementary to thymine? That's adenine. Guanine is cytosine. Which of the following? It's gonna be GC. What is the term? It's semi-conservative because only half of the original is original, the other half is new. It's A to T. What process is involved in the unwinding of DNA? Okay, unwinding of DNA and breaking of H bonds. That is DNA helicase. CCC, okay. What type of reaction joins adjacent nucleotides? Condensation. So we should have mentioned condensation. Via condensation reaction. Each molecule of a newly synthesized DNA contains half the original DNA material. Which protein is involved in the condensation? That is, again, DNA polymerase. How many DNA polymerase molecules are needed per DNA molecule? 2. Wait, per molecule. Two per molecule? Those are 2 separate molecules, 2 per molecule. Right? After DNA replication, what is each DNA molecule? No, it's 1, I'm pretty sure. Yeah. After DNA replication, what is each DNA molecule comprised of? 1 original and 1 new polynucleotide strand. DNA replication is during interphase. A requirement. I'm pretty sure it requires ATP. DNA helicase unwinds 2 strands of DNA by breaking the hydrogen bonds linking the complementary base pairs. I'm expecting 80% maybe. Oh yeah, 95 will do as well. Okay, so, couple of things to note before we move on to RNA. Number 1, DNA helicase unwinds the DNA molecule because it's breaking the hydrogen bonds. Free-floating nucleotides will bind to their complementary base pairs. Yep. DNA polymerase joins adjacent nucleotides by forming phosphodiester bonds via condensation. Okay. We're good to move on to RNA, I think. Or should we do a quick quiz on DNA? Honestly, I forgot how easy DNA is as a topic. That's taken me 20 minutes. I'm just grabbing a drink. Okay. Let's see. Okay, I think we're ready to go on to RNA. RNA, ribose nucleic acid. The idea here is the structure has ribose. It has, I'm assuming, the phosphodiester, the phosphate group, and a complementary base, a nitrogenous base. Now, the interesting thing about RNA is the bindings. A, T, C, G. Adenine will bind to uracil. Thymine will bind to adenine. Cytosine, guanine. Guanine. Sorry. That's a terrible C. So, already, that is the structure of RNA. This is obviously a ribose. That's going to have a phosphodiester bond. The idea, though, is that this is a single molecule. Okay, RNA is only 1 strand. It is a smaller strand. It has different base pairings. It has uracil instead of thymine to bind to that. So in an exam, I have seen this numerous times where they will ask you, contrast the structures between DNA and RNA. And people will put, people will waffle, okay? Just a sec. Okay, people will waffle at this question. What are the differences in structure between DNA and RNA? Number one, look at the names. RNA, not, DNA. The difference there is the sugar. DNA is deoxyribose, RNA is ribose. Second one, the molecule is a single strand, unlike DNA, which is a double strand. Okay, this is like RNA, DNA would be like that. The second idea, RNA is a lot smaller of a molecule because it's only a part of a DNA strand being copied via transcription. So it's a smaller molecule, has ribose strand. It has uracil. Uracil binds with adenine instead of binding with thymine. And that is everything. So that right there, we're already done with RNA. Just have to know how different it is in structure-wise. Let's go on to the evil part of this topic, transcription and translation. Now, I do not recall this very well, so I'm going to be completely honest with you guys. I might get a lot of this wrong. There is only one way to find out, and that's by dumping it on the board first, doing a quiz to see what we can get right with our knowledge. So, it starts off almost identically to semi-conservative replication of DNA. We have our strand of DNA. And this happens in the nuclei. So bear that in mind. Nuclei. Okay. So, just like before, we are saying that the first step is DNA helicase causes the bonds to break, breaks hydrogen bonds, causing the molecule to unzip, is it unzip or unwind? I always get the two mixed up. Anyways, DNA unwinds. That is the first step. The second step is the free-floating RNA nucleotides bind to one strand of the DNA. So in other words, only one acts as a template. Only one strand of this DNA is acting as a template. The other side can piss right off. Okay? So we have our RNA. I am butchering this drawing. Oh my god. So they are going to bind to their complementary base pairs, but only on one side. Unlike DNA replication, where they would bind to both sides. So that is a huge, right there. That is a giant thing you need to get in your heads. In DNA replication, both strands act as a template. In transcription, one strand acts as a template. That is the difference between like 3 or 4 marks, to be honest. It's that big. Okay. So, now what we have is something like this. Like that. Okay? So, we are then going to say that RNA polymerase is it RNA synthase or polymerase? I'm going to say polymerase forms the phosphodiester bonds via condensation between adjacent RNA nucleotides. So, Then what we have is this strand of RNA. Before it leaves the nucleus, the fourth step is that the introns are removed and entrons, is it extrons? Entrons, I don't know. Extrons are spliced or fused together. I'll just say extrons are binded together. Remember, introns are non-coding DNA. Introns are only present. Introns are only present in eukaryotes. So, I believe that is transcription right there. Oh, and then it obviously... it leaves the nuclei. So that right there is the entire idea of transcription. Very simple to understand, almost identical to DNA, except for a couple of simple changes. And I don't think you need to talk about the idea that these will then bind back together. So, that is the idea for transcription. But how do we turn this RNA copy, this mRNA copy, I forget, before it becomes mRNA properly, it needs to, be spliced or whatever, pre-mRNA. That's the term right there for when the introns are still present. mRNA is what you use to refer to it after splicing has occurred. So, final thing we're going to do now is translation. The idea here is this occurs in the ribosome. Firstly, we need to talk about tRNA, which I don't recall the exact structure of. I know it kind of resembles like... I don't know how it's meant to look like a clover leaf, okay? Please bear with me, guys. I did not do GCSE art. The structure of this, though, you can easily talk about. You can talk about this being the anti-codon. You can talk about this being the amino acid binding site. Or overall, you could talk about its clover leaf shape. Do not care what you talk about here. But in an exam, if you get asked to describe 3 features of a structure, you know what they are, okay? And this has happened before. There have been exam questions where you have to discuss the structure of this. Make sure you do. So, let's talk about it. I'm going to draw a ribosome like a burger because I think that it actually kind of helps here, okay? So that's the ribosome, okay? Ignore how goofy and janky this is. Please bear with me, guys. Again, I did not do GCSE art. The idea is that this is intended to be it looks like a face now. The idea is that only one triplet is gonna work, will be read at a time. Okay? Only one triplet is being read at a time by the ribosome. And the idea is that the triplet will... Okay, now I'm really struggling to word this. Okay, the RNA strand will be fed through the ribosome. Each triplet will be read. And for each triplet, you have, obviously, your tRNA brings the complementary amino acid. And via a condensation reaction catalyzed by, I think, ATP nearby, condensation occurs to bring the bonds together. So I'm going to need a look at this because the idea is really simple, like I've just drawn. It's literally just the RNA strand goes through the ribosome, one triplet at a time is read, that triplet is then passed through. It's a bit like a conveyor belt. I wouldn't know how to word this first, so I'm going to go all the way back on our old Google Classroom, and I am going to find out what the teacher said. Fuck blind people. And deaf people suck. And so do cripples. Dumb quadriplegic fucks. Okay. mRNA attaches to the ribosome. Each tRNA has a complement. Yeah, so forgot to mention the anticodon will obviously bind. One thing I've misdone in my drawing here is overcomplicate it. Let's try and undercomplicate this sketch so that I can really show how easy this is and how difficult I'm making it. Let's literally draw the ribosome like a square. Because, again, trying to make it look better is just not working. We're going to draw it to be like this. Okay, so 1, 2, 3. So what will happen is the tRNA will bind to the codon. Its anticodon will bind to the complementary codon, and it will bring the complementary amino acid via the binding site. So let me just... The first tRNA forms hydrogen bonds. Second tRNA bonds with the next one. Peptide bond forms. I'm 90% sure I've got everything then. I'm 90% sure that I've got everything. It's literally just RNA moves through, it attaches to the ribosome, okay, number 1, attaches to the ribosome. The first triplet is read, the complementary anticodon will bind to the codon. This tRNA has, obviously, this specific amino acid, and the more this occurs, hydrogen bonds will form peptide bonds between the amino acids. Very convoluted way of basically saying that shit happens, okay? Right. Let's go into protein synthesis quiz. Oh, there's only a transcription quiz. Okay, well we could do this and then end the stream. What type of molecule is a transcription factor? What the fuck does what the fuck does that mean? A factor? Polypeptide, maybe. Right, okay, we have not done any of those, so I'm gonna immediately back off. Okay, there are no quizzes whatsoever. What about DNA within cells? We could do this because it's been a while. A thread-like structure of nucleic acids and proteins found in the nucleus of most living cells. Thread-like structure. Is that literally not just chromosomes? The names of the proteins that the chromosomes are wrapped around, DNA is wrapped around, are histones. A section of DNA that code for specific polypeptides, yeah, we already know that. That is a gene. Each human chromosome is comprised of one DNA molecule. It's 2 DNA strands though, no? So that's false. End of a chromosome is called a locus. Specific terminology for the position is a locus. Hold on, so what's the end of a chromosome called? The telomere? DNA replication occurs during interphase. Type of chromosome are found in eukaryotic cells. Both. You have circular DNA in the mitochondria and chloroplasts, and you have linear DNA in the nuclei. What type of chromosome are found in prokaryotic? It's only circular. Prokaryotic and eukaryotic use the same bases, yes, because they're universal. The same bases code, well, the same codon codes for the exact same amino acid. Prokaryotic DNA molecules are wrapped around proteins. No, they don't have histones in prokaryotes. They supercoil. What is the name of the extra molecules of DNA found in some prokaryotes? Plasmids. Name of a pair of chromosomes that have the same genes in the same loci. Homologous. No idea if DNA has a negative charge. I guess it would make sense. I'll put true, but it could be wrong. It might be neutral, actually. If DNA is stable, why would it have a charge? What do you inherit your mitochondrial DNA from? Both parents equally. Mitochondrial DNA is not linear. Mitochondrial DNA is evidence for evolution. Chloroplast contains circular DNA. True. I'm going to say 80% for this one. 72%. Oh, which ones did we get wrong? Each human chromosome is comprised of only one DNA molecule. That's true? Hold on, are we... Maybe I misread that as DNA strand. I guess the overall DNA molecule is 2 strands. Okay, so I misread a question. Type of chromosomes are found in eukaryotic cells. That is wrong as well. I would have got that right. I said circular and linear because circular DNA can be found in multiple organelles. Not just linear in the nuclei. That's wrong. They do have an overall negative charge. Did not know that. You inherit your mitochondrial DNA from your mother. Okay, I must have misread that as you inherit your DNA. Cool, okay, so I got about 75%. Would have done better if the questions were right. I'm honestly done. That has been a good 45 minutes of revision. I am happy to end there. Plan for the rest of the day. I do need to get my personal statement improvements done because we are running out of time on that. But plan for the rest of the day. I will make 1 or 2 quizzes about transcription, about translation, about every topic we've covered in the last week. Let me know how well you're doing that, and we'll get back to it next week.